At the end of your last PowerPoint, I posted some review questions. I did not mention you had to complete these. However, for those of you who did go through these, let's go over those answers. Okay, number 1, which of the following is or are true concerning the atomic nucleus? Let's first draw out our nucleus and what we know the atomic nucleus before we answer the question. So here's our atomic nucleus. What do we have in here? Well, we have the protons and we have the neutrons. What charge do we have on protons? Good, positive, P for positive. Neutrons is N for neutral. So there's no charge on that. Surrounding this, in our shells, we have the electrons, which bear a negative charge. The letter A contains the mass of the atom. Well, what do we know gives rise to the mass of the atom? We know that protons plus the number of neutrons will give us our mass. And so these are found within our nucleus, so letter A is correct. Letter B, the negative charged particles are here. Well, we just went over that. Those are found here within our shell, so that is incorrect. Particles can be ejected. Okay, this one, under normal conditions, if we have a neutral atom, meaning we have equal number of protons and neutrons, then we don't have any ejection of our neutrons. However, if it's a radioisotope, then we can eject those neutrons. And I mentioned that in the last video, especially if you have the radioisotope of iodine. Right, so this one is kind of half and half. Contains particles that determine the atomic number. What specifically defines our atom? Good, that's going to be our protons. So this is yes. And contains particles that interact with other atoms. Well, when it gives us our bonds and our interaction between other atoms is the electrons, whether we have enough electrons in our outermost shell or if we just want to donate that. And so this has to deal with electrons and not our nucleus. Great. Number 2, pick out the correct matches of element and number of valence shell electrons. Oxygen being 6, chlorine 8, phosphorus 5, nitrogen 3, and carbon 4. For this, let's go look at the periodic table, and actually let me first cover what exactly a valence shell electron is. Anytime, so here's our nucleus, here's our first shell, here's our second shell. First one has how many? Good, 2. Second one has up to 8. But now let's say in the instance that we don't have 8 in our outermost shell, we only have 4. This atom will have a valence shell electron of 4. So it has 4 valence shell electrons. And so those are the number of electrons in the outermost shell if an octet is not complete. Let's look at this periodic table. Okay, let's look first at number 1 example. That's going to be oxygen. And our question asks us if oxygen has a valence of 6. All right, so first what we need to do is look at is oxygen. Here we have 8. And so we have 8 protons. And therefore, because this is a neutral, we're not talking about anything that's charged. This also should have how many electrons? Good, 8 electrons. Let's draw our nucleus. First shell should have how many? 2. Second shell should have how many? 6. 2, 3, 4, 5, 6. All in all, did we have our 8 electrons? Yes. 1, 2, 3, 4, 5, 6, 7, 8. We're good. In our outermost shell, how many electrons do we have? Well, we have 6. So here we have 6 valence electrons. Are we right? We are correct. So number 1 is correct. Number 2, it's asking if chlorine has 8 valence electrons. Chlorine, we have 17 protons, therefore we have 17 electrons. Drawing our shell. First one, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Next shell, we can carry at most 18, but how many more do we have left? 7. 1, 2, 3, 4, 5, 6, 7. How many do we have in our outermost shell? we have 7. And so this number 2 is incorrect. Number 3, let's go over here. Phosphorus. Does it have a valence of 5? Let's look for that over here. 15. So 15 protons equals 15 electrons. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And the outermost is 5. 1, 2, 3, 4, 5. Good. And so valence of 5 and 5. That's good. All right. And so you can do the nitrogen and carbon on your own as practice. And a great way to check also if you're doing this correctly, and this is something you'll probably learn if you take a chemistry class, is that if you look at the periodic table, we organize the periodic table in what are called groups and periods. So going from left to right, these are periods. Up and down, all these columns, these are going to be groups. And really, whatever number the group is in, or whatever number the column is, that's how many valence electrons these atoms are going to have. So for example, hydrogen, because it's in group number 1, it's going to have a valence of 1. Beryllium, because it's in group number 2, is going to have a valence of 2. Boron, number 3, so it should have a valence of 3. All the way to helium, which is should have 8 in its outermost shell. Let's take a look and double check what we saw over here. So oxygen, it asks us if it has a valence of 6. Let's look at oxygen. 3, 4, 5, 6. It's in group number 6. And it is consistent. We have 6 valence electrons. Chlorine, does it have a valence of 8? Let's go for chlorine. It's in group number 7. So we already know it's not an 8. And we are consistent with our findings. Phosphorus, 5. Here's phosphorus. We are in group number 5. We are consistent. Okay. Quick check. Number 4, nitrogen is asking, does it have a valence of 3? No, nitrogen, we are in group number 5, so we already know that's going to be wrong. 5, carbon, does it have a valence of 4? And we are correct. Okay. No, number 1, 3, and 5 should be true. Those are all correct. Okay, number 23, the 2 oxygen atoms forming molecules of oxygen gas that you breathe are joined by a polar covalent bond. What do we know so far about polar covalent bonds? This means unequal sharing of electrons. And explain why this statement is true or false. Here we have oxygen. Oxygen has a double bond. It's the exact same type of atom. 2 lone pairs of electrons in that. And because this is the exact same type of atom, they both have 2 lone pairs of electrons, same charges, same bonds, we know that this isn't a polar covalent bond, it's actually a non-polar covalent bond. Okay, and we just gave the reasons why. Last one, identify each of the following reactions as a synthesis, decomposition, or exchange reaction. This first one, what do we have? We have a synthesis. Number 2, what do we have? We have an exchange. Okay, so we have iron and sulfate coming together, and then copper is going to be on its own. Number 3, what do we have? We have an acid, strong base, forming a salt and a water. This is going to be, what is this going to be? This is going to be an exchange. But a specific type. Okay, so hydrogen is going to go to OH. Sodium is going to go to Cl. Is a specific type of reaction called a neutralization reaction. And then finally, our last reaction, what do we have? We have a decomposition. Great. And you should be able to go through these questions and feel comfortable answering them. Okay, now let's finish off chapter 2, looking at basic chemistry. And this chapter we're going to go through all the different types of organic compounds and different categories of them. We're going to first start off with the carbohydrates. Now carbohydrates will always be found in a carbon-hydrogen-oxygen ratio of a 1 to 2 to 1. They are classified according to their size, where monosaccharides are the smallest type, and therefore they're called simple sugars. The most popular one we know is glucose. Disaccharides, these are two monosaccharides joined together. And we know this because of the prefix di, and so a lot of times you'll see sucrose, lactose, maltose. These are a couple of examples. And then finally we have polysaccharides. Poly means what? Many. And so these are long chains or many monosaccharides. The most important one that we're going to be looking at and talking about is glycogen, which is a storage form of glucose. So here's that monosaccharide, double or two sugars coming together, disaccharide, and then finally your long chain. So it can be a nice long chain. You can also have branching, so coming off of that. Here are the common carbohydrates that we have in our body, and why are these carbohydrates important? Well, we use them as an energy source, and we'll be looking at how glucose, these carbohydrates, can be used for what's called aerobic respiration, and how it's a quick, fast way of just getting energy. Alright, so glucose, all these carbohydrates, what's also important to note is that we can't produce them in our own body, so we always have to have it from a plant source. Okay, so glucose, fructose, some common monosaccharides that we know of. Disaccharides, sucrose, lactose, and maltose. Lactose specifically present in our milk. And polysaccharides we just mentioned, glycogen being the most important one. Now starch, also another type of glycogen, but this isn't the storage form that we have in our body. Glycogen is a storage form that we have, so anytime you consume a large meal, you store our glucose as glycogen. Starch, this is a polysaccharide found within plants, so it's very different from our bodies. This should look familiar. How do we take these monosaccharides and then form them into disaccharides or into polysaccharides? What do? We remove two hydroxyl groups, and this is, well, not two hydroxyl groups, I'm sorry. We take one hydroxyl group and we take a hydrogen. We remove this. This is called dehydration synthesis. And then going the other way, anytime you want to utilize this glycogen or this disaccharide source, then you need to break this down because our body can only utilize the monosaccharide form. And so to do that, we're going to go the opposite way, which is called what again? Hydrolysis. Anytime you see hydro, that means water. Lysis, this means to split or cut, so splitting with the use of water. Lipids, this is the next category of organic compounds that we're going to be looking at, and this is a ratio of carbon and hydrogen is going to be greater than oxygen. Are these soluble in water? And you've probably seen this already if you tried an experiment at home whenever you wash your dishes. Anytime you add water to lipids or fats, then they cannot mix with each other, right? So they're not soluble at all. So they are not soluble. And we're going to get into the reason why that's the case. Common lipids that we see within the body, we have triglycerides. This is the one that we mostly know. Eat too many malasadas or drink too many milkshakes. Neutral fats. Triglycerides. Well, I guess not really milkshakes in Hawaii, right? We eat the shave ice. So this is found in your fat cells, which we call adipose tissue or adipocytes. So adipocytes are specific fat cells. And we have fat tissue, which we call adipose. Second area where we find lipids, phospholipids. Second type of lipids, phospholipids. And we find these within cell membranes. Now I'm going to quickly draw this. Here is our cell. We have a nucleus within our cell, and the ability to separate the external environment from the internal environment, you need this barrier, and this barrier is called that cellular membrane. And so that's composed of these phospholipids. Here you'll see we have this head and then also tails. Next one we have are steroids. Couple of examples of steroids that we have within our body. We have, oops, sorry, I dropped something. Okay, so we have vitamin D, cholesterol, vitamin D needed to help store your calcium, hormones, and cholesterol isn't always that bad because there's some good cholesterol which we need for our cell membrane. Now let's take a look at each of these lipids one by one, starting off with the triglycerides. And even though we might not think of fat as being the greatest thing within our body, it actually is one of the greatest sources of yielding a high amount of energy. It's composed of a glycerol, backbone, and 3 fatty acid chains. And so here's what that looks like. Here's that glycerol. You have 3 fatty acid chains. Here's 1, 2, 3. And how do we form this to form that triglyceride? We're removing water, so this is dehydration synthesis. And so in the process you give off water as a byproduct. Now, what other things are great about these triglycerides? It's absorbed from your food. Or synthesized in your cells. And so sometimes if you absorb a lot from your food, you can break it down and store it, but then resynthesize it once you need it again. So remember that concept of homeostasis. And anytime we need it for other regions of our body, we can transport this to our blood for use in other cells. There are 2 common types of triglycerides. There are saturated fats and we have unsaturated fats. Now saturated fats, these are the bad types of fats because they increase bad cholesterol. And when we look at digestive system, we'll see exactly cholesterol in itself. Why is it good? Why is it bad? It is associated with atherosclerosis, and heart disease. Okay, and atherosclerosis, if you look at your vessels, what happens is that really what this does is increases cholesterol and buildup, plaque buildup here within your vessels. And over time, what does that do? Here, if this is a cross-section of your vessels, it starts reducing the diameter and ability for blood to flow through. So eventually, sometimes if something gets stuck, like a clot gets stuck over here, more red blood cells in turn can get stuck, and eventually you can cut off blood supply to lower organs and extremities. Examples of this, so this is coconut oil, whole milk, cheese, oh darn, it's so good too, right? And vegetable oil. Why exactly do they call it saturated fats? Well, it's because these bonds are composed of single carbon-hydrogen bonds. Now if we look here, we have the glycerol backbone. Here's that fatty acid chain. Anytime you see something like this, when you look at organic chemistry eventually, these points, these are the carbons. Okay, and attached to these carbons we have hydrogen atoms. Alright, and so these are long chains of hydrocarbons. Let's go back over here. Okay, so here's what that looks like. Long chains of hydrocarbons. Here it looks like the hydrogens are next to each other, but in actuality, you have hydrogen up here, hydrogen up here. Okay. Alright, so these are long chains of those hydrogen carbons. Why is this bad? Well, if you can think about butter, at room temperature butter is solid, and if butter being a saturated fat, let's look at the structure right over here, and if you look at it, it's pretty linear. All these fatty acids can stack right on top of each other. And I actually outlined it right over here. It kind of looks like a block. So if you take one saturated triglyceride, so here one triglyceride, you take another triglyceride, what eventually happens? They stack very nicely on top of each other, which is why at room temperature they have this nice stacking appearance. Now this is very different from unsaturated fats because these are not saturated with CH2 bonds, meaning that we actually can create carbons that are double bound to each other. So here is carbon-carbon. Here you have a hydrogen, 1, 2, 3, 4. Here you have a hydrogen. And so we have one less hydrogen at these carbons, and you can create double bonds. And when you look at it from a faraway distance, what happens is now you can create a kink in this chain. And so is it going to be easier or more difficult for these to stack up at room temperature? More difficult. So I'm going to try to outline this shape over here. Let me clean that up really quick. So here's that shape that we created. And if you think about how these are going to be at room temperature, they don't stack up very nicely. And because of that, they're going to take on this liquid appearance. So unsaturated fats, they have at least one double bond. Between the carbons in the hydrocarbon chain. And these increase good cholesterol. A couple of examples, we have avocado and olive oil. Triglycerides, for us to use these, what needs to be done, we need to break down this triglyceride into its precursor form. We got to break them down into both our glycerol and into fatty acids as energy sources. And just like what we saw earlier with sugars, you remember you have your monosaccharide, you have your disaccharide with the O, and you also have your polysaccharides. We can't utilize a polysaccharide when it's bound. Okay, we have to break it down to its monomer form. Same thing with our disaccharides. So it's the exact same concept carries over to these triglycerides. We need to break down the glycerol and break down these fatty acid forms. And then these can enter into our cells very differently and then be utilized for an energy source. Phospholipids location, these are going to be the most abundant type of lipid found within our cell membrane. So location within our cell membrane. These are diglyceride. What does that mean? We have the glycerol backbone, but instead of having 3 fatty acids, we only have 2 fatty acid chains, and we have this phosphate group on the other side. And so because of this, we have what's called phosphate groups. These are negatively charged species, and because of that, we have what's called a polar head because of that negative charge, and a phobic tail. Now, what exactly does that mean? So hydro means water again. Phobic, if you have a phobia of something, you have a fear, so it's scared of water. So anything that's hydrophobic does not want to be close to water. Now, why does this make sense? Let me clear this off really quick. And remember water. Water has a partial negative, partial positive charge. Phosphate group, I just mentioned, has a negative charge towards that. So if you put a phospholipid in water, what is going to be closer to your water molecule? The phosphate head, right? And here we have partial positive, partial negative. Opposites attract. I'm going to just draw a generic version of this. This is a phospholipid. This is partial negative charge. And so this is going to have an attraction. Now, hydrogen and carbon, are these charged? Well, they're not, right? If you look at this, these are considered to be covalent bonds. And covalent bonds do not have that nice, well, this type of covalent bond don't have that nice attractive pull towards it. This is equal sharing of electrons. Carbon doesn't want to pull hydrogen closer towards it compared to oxygen and hydrogen. And because of that, these are going to not have any charge with them. And since there's no charge, it's not going to want to interact with something that is charged. And so like will always dissolve like, so this is going to be closer together. And anything that's not compatible, if there's no charge, it also wants to be something with no charge. Okay, so this is why in water and the cell membrane, where we're going to learn in the next chapter, I just want to give you some background information about it right now since you're seeing it. All of these phospholipids are going to organize and orient themselves so that all these fatty acid chains are going to be facing the inside. All of these charged portions, these polar heads, are going to be closer to water. And eventually, this is why, if you've ever heard about our cell membrane, it's called a lipid bilayer. So lipid, because it deals with a phospholipid, bi meaning two, layer. So here's our first layer and here's our second layer. And that's really what gives rise to our cell membrane. Now lipids, another type we have are steroids. Steroids are flat molecules formed of 4 interlocking rings with the same framework. Here is that first ring, second ring, third ring, and fourth ring. Again, anytime you see these points, by default, these are going to be carbons. Okay. An important steroid that we have is cholesterol. And it's because it serves as a basis for all the other steroids made in the body. Where else do we find this? We find it in bile salts. And this is going to be for part of the digestive system. And vitamin D. And some hormones. Okay. And once again, we can also find some of these steroids as a component of our cell membrane. Next category, we're going to look at our proteins. And proteins account for over half of the body's organic matter. And because of that, they play a vital role in cell functioning. There's actually over, or about 100,000 different types of proteins in our body alone. And why are they so important? Because they actually create and they produce a lot of the chemical reactions and initiate a lot of the chemical reactions in our body. So these are made of building blocks called amino acids. In our body, we have 20 in all, or there's known to be 20 in all. Not all of them are produced by our body. Some of them we actually get from our diet. And these amino acids can come together, so I'm just going to give the abbreviation amino acid. We're going to come together to form chains of amino acids called polypeptides. And what are their functioning? They function as enzymes, hormones, antibodies, lots of functions within our cells. If we look at an amino acid, this is a general anatomy of one amino acid. And here we have an amino group, so N and 2 hydrogens attached to that. We have our central carbon, which everything stems off of. Carbon will always have 4 bonds to it. Here we have a hydrogen. So all of them are going, all these amino acids are going to have a hydrogen side group. And then we have a carboxylic acid group. So C, double O, and then OH. And then what defines and gives a unique identity to each amino acid is the R group. So gives to T of the amino acid. All right. Here is an example of other amino acids found in your textbook. Here's a general structure of one amino acid. Here's your central carbon, carboxylic group, amino group. Here's one type of amino acid called glycine, and the R group is actually a hydrogen, so it's the exact same as this one. Aspartic acid, here we have our carboxylic acid group, and then here we have a CH2. Lysine, we have another amino group, and we have a longer hydrocarbon chain. And then finally, cysteine, one of the only ones that have sulfur, so S is for sulfur. How are these amino acids joined together? Well, we see this removing water, so what do we know? Dehydration synthesis. Okay. And so this is how we can form longer chains of amino acids called polypeptides. Once again, we can also use these amino acids in our body. So once you consume, for example, chicken, right? You consume meat. Okay, here's a drumstick. You're going to break this down, composed of lots of proteins, lots of these polypeptides. We're going to undergo hydrolysis, break these down into smaller building blocks. And then, so this is dehydration synthesis, and this is a catabolic reaction because we're going from polypeptide to amino acids. Now, what can we do? Now that we've all these building blocks, as a review from that first chapter, what can we do? We can build this up to something else. So we can build this up to enzymes, proteins within our cells, lots of different functions. And that's done through what process? Anabolism. So hopefully things are starting to come together. I know it's kind of a lot of material. Now, let's look at this, how proteins are organized. And we looked earlier, amino acids. Here's one amino acid. Here's another amino acid. Okay, and they're organized by formed into polypeptides by dehydration synthesis. And they're not going to stay in this form. Remember, we have all these hydrogen atoms coming off of those amino groups and carboxylic groups. So what can hydrogen atoms do? Hydrogen bond. Okay, so because they can hydrogen bond, and then also some of these amino acids, they like being close to water. So what term do we give that? Hydrophilic. And some of them don't like being close to water. What term do we give that? Hydrophobic. And so some of these amino acids are hydrophilic, some are hydrophobic. Some of them can hydrogen bond, and some of them don't even want to bother. And because of that, this first primary structure, this first polypeptide, can then fold and to form another three-dimensional shape. So we're going to start off with this basic, most organization of this polypeptide, and that's called a primary structure. So primary structure, all that is just the sequence of the amino acids in a chain. And because they can interact, that's when they can form secondary structures. And there's two different types that we're going to look at. This is a alpha helix, and you can notice is a nice helical structure that's being formed. Okay, so helical structure. Another thing that can form is a beta-pleated sheets. As a review, and here's this hydrogen bonding. And remember, oxygen, partial negative, hydrogen, partial positive. They have this nice attraction, and because of that, it's going to pull each of these compounds. So here is, okay, wait, where's my essential carbon? Carboxylic carbon, nitrogen. Okay, here's one, here's another. We can form, each amino acid is actually going to be called a compound. So each of these compounds, and we saw this before, each of these compounds can interact with each other by these hydrogen bonds, and that's going to be that intermolecular bond, because it's between two different molecules which are specifically called compounds. Okay. Okay, I'm going to kind of go back. So here is our long amino primary sequence over here. And as you can tell, because some of them are hydrophobic, some are hydrophilic, some of them like forming these hydrogen bonds and in turn form these alpha helices. You can also form these beta-pleated sheets. And so you'll notice, because these are intermolecular bonding hydrogen bonds to form these beta-pleated sheets, what can happen is one portion of your amino acid sequence over here, it can undergo a turn, come back, and then interact with this first portion over here. So it doesn't have to be directly next to each other. It can be far away. So here's portion 1, here's portion 2. The turn is number 3. Come back around, this long portion over here is number 4. So number 2 and 4 can interact with each other to form those hydrogen bonding and form those beta-pleated sheets. Now, if you collectively put all those secondary structures together, so all these alpha helices, these beta-pleated sheets, your primary structure, hydrophilic, hydrophobic interactions, this forms your tertiary structure. This is only from one polypeptide. Okay. Now, let's say that you have multiple polypeptides. So here's one polypeptide. Here's another polypeptide. Here's another polypeptide down here. And then here's your last polypeptide. And the green one we can say is going to be this one over here. And so of course you make 3 more of those. These in turn can interact with each other, come together to form this larger unit, and this is a complete protein. And this is called a quaternary structure. Okay, so this is a very final 3-dimensional conformation, and these are the structure is the one that carries out all the different reactions within our body. Alright. Here is this overview once again. Primary structure, just your simple amino acid sequence. There's no interaction between the different amino acids. And then here you have hydrogen bonding causing the twisting and turning of your entire amino sequence. Here we have one region comes in. You can have a bend, come back over here, and then you have interaction, hydrogen bonding between 2 different regions of your polypeptide sequence. And then finally here, tertiary structure, you take all of that together, and what do you get? So here's just your long sequence, you're bending it, folding it, and then finally you form your quaternary structure. Okay. This is just an illustration or an image of all the amino acids utilized by our body. You do not need to know this for an exam, but just to kind of give you an idea of some being nonpolar, polar, basic, or acidic. Okay, quick review. R group, that's the one that's going to be interacting with our neighboring amino acids and also interacting with our solvent. So if it's going to be, let's see now, here's our R group. Okay, so here, this is nonpolar, no charge on this, so it's going to want to be away from water. And so typically in a protein, here's your 3-dimensional structure, it's going to be found somewhere here, hidden from the external water. Polar, these ones you'll probably see closer to the surface. Over here. Those wants to be closer to water. And so that, once again, will determine your final structure. And once that happens, your final shape will determine the function of the protein. How does fever affect this? Okay, thinking about homeostasis, we don't want to have high fever for too long, and that's because heat, acids, bases, they can all denature or remove this three-dimensional conformation of our protein. Here's our protein under normal conditions. It can perform whatever action or whatever it's going to carry out. If you have a high fever, it's going to break some of these bonds that we've created. Acid also can start breaking some of these bonds. And now all you have is this polypeptide sequence of your amino acids, and there's no function to this. It's non-functional. And that's dangerous, especially if you're dealing with proteins, if they want to help for your immune function and also help break down all those carbohydrates and help for your metabolism. So effects of fever, these will denature or deform the three-dimensional conformation of your protein. Okay. One important thing about these proteins is the fact that they can serve as catalysts. And what catalysts are is that they increase the rate of chemical reactions without themselves being consumed. Meaning that they're not going to be part of the products that they're formed, nor will they change their function. Nor will they change at the end of this entire reaction. Okay, so another thing that they do is they help hold the substrate in place. Lots of terminology here, right? Okay, here is our enzyme. Substrates, these are the ones, kind of like you think of the reactants. These are the ones undergoing the reaction. Active site, these are where those reactants or substrates will bind. And if you think about a normal cell, if we want to combine amino acids together, here is our huge cell. What happens is just by kinetic random motion and energy, these amino acids can come together and then form. Specifically when we build these polypeptides, we kind of have to call them in. And so it's random kinetic motion that's going to eventually form into this polypeptide. Now what enzymes do is that they house an area specific for the substrate so that it reduces the amount of time for the reaction to take place. So imagine this is all by chance alone. They're going to come into the correct conformation and correct reaction. But here we have a seat just for it to kind of come in, sit down. Now that we're in the correct orientation, we can create these bonds much faster to produce our product. Okay, so here we want to produce a dipeptide. So enzymes are very important for those reasons. Next type of category of organic compounds we have are high energy compounds called, we're going to look at specifically adenosine triphosphate or ATP. And what type of energy is this again? This is that chemical energy, and we store that as potential energy. So potential energy is contains the ability to eventually, once those bonds are broken, perform mechanical work. So stored potential energy in bonds. And ATP is needed in order to drive a lot of these reactions. Now, how do we generate that? The easiest, fastest way to produce all this ATP is by glucose. It's the most important fuel for your body cells. And glucose itself, if you take one glucose molecule, it's not automatically going to produce ATP. It's actually going to enter into our cell, work under specific organs, which we call organelles, and that's going to start breaking it down and utilize that and harness molecules of ATP over time. So everything in order for us to gain energy, all of that has to go into our cell. Okay, so here is our adenosine. This is that backbone. Phosphate, so adenosine triphosphate for 3 phosphates. 1, 2, 3. Breaking these bonds release lots of that energy to drive mechanical work. Okay. Examples of where we see this mechanical work. Here, if you want to take your substrates and then form a product, you're going to take 1 or 2 reactants. You can use this phosphate. Once this bond is broken, remember it's a high energy bond, it's going to release that. As this releases, you're going to release energy. This energy is going to allow these 2 reactants to come together to form your product, and then you form ADP as an end product. You saw this in the last video, this ATP, sodium, potassium pump, needing ATP. In order for solids to go from one area to another, and especially if it's against its concentration gradient, we're going to need ATP for that. And then finally, for contraction of your muscles, both your smooth muscles and your skeletal muscles. So smooth muscles, where do you find this? You find this in your vessels, your digestive tract. Okay. And then finally, our last compound, organic compound we're looking at, are nucleic acids. And nucleic acids, these are important because they store genetic information that provides the blueprint of life. How you came to be uniquely yourself, your character, physical, genetic characteristics, how that's unique compared to your siblings or your parents, all that's because of your nucleic acids, specifically your DNA. Now, it also provides information used to making proteins. That's the specific role of your DNA. So there's 2 different types of nucleic acid categories we have. We have DNA and RNA, and that stands for deoxyribonucleic acid and ribonucleic acid. And both of these are named after the sugar that forms the basis of the nucleic acid. Okay, so named after the sugar used to produce the nucleic acid. DNA, you'll see this typically as a double helix. And the helix was that helical, was that slinky-like structure. It has 2 right next to each other. That's a double helix. And then ribonucleic acid, this is a single helix. What is it named after? These sugars, and both of these sugars are pentoses, meaning that they are they have 5 carbons to them. So 1, 2, 3, 4, 5 carbons, forming this ring. Here we have on our second carbon a hydroxyl group, and this defines the ribose sugar, and that's really what we find in our ribonucleic acids. However, in deoxyribonucleic acid, on our second sugar, we only have a hydrogen. So it's the D, meaning without that extra oxygen area, the oxygen atom. So deoxyribonucleic acid has this type of sugar, the deoxyribose. Okay, let's compare and contrast the differences of the sugars and then also how these nucleic acids are formed. Okay, so the anatomy of one nucleic acid, they will always have a phosphate. Here is that phosphate. And here you can notice those negative charges on those phosphates. It'll have a sugar and it'll have a base. So here's your base, sugar, and phosphate. So it always, nucleic acids, whether it's RNA or DNA, will always have these 3 components. Now, what's going to differ is going to be the sugar. So DNA is going to have deoxyribose sugar. RNA is going to have the ribose sugar. And another thing that's going to differ are these bases. And these bases, in DNA, you're going to have cytosine, guanine, adenine, and thymine. And then in RNA, you're going to have uracil, adenine, cytosine, and guanine. And we'll write that down in a little bit. Okay, so where do we find nucleic DNA, so deoxyribonucleic acid? We find this in the nucleus of the cell. Here's our cell, right in the middle. We'll find all of that DNA. Sugars, what sugars did we mention already? We had ribose, and then bases, we had A, T, and then C, G. Now, in high school, I used to be in color guard, and the first time I learned this, I remember thinking to myself, AT is at color guard, CG, and that's really how these two nucleic acids are going to eventually bond and hold together. And so you notice here we have this double helix, and this first sequence of the double helix, or the first strand of it, is going to have either your A, T, C, G, or whatever it's going to be. And then on the opposite side is going to be the complementary strand. So that's what I'm talking about as at color guard. So T is going to be complementary to the A, G is going to be complementary to the C, C and G complementary, and then A and T complementary. Okay, so back and forth. And here in purple, what this is that deoxyribose sugar. So this is what defines DNA. And then here in pink, this is that phosphate. And so both your sugar and your phosphate form this backbone of your entire double helix. Interaction and holding this double-stranded DNA together is going to be all of these bases. Okay, and so if you notice, if we go back, do we have the structure? We don't have the structure. Okay. So really what holds these together, here's a structure, are hydrogen bonds. Here's thymine. Here's your adenine. We have oxygen, partial negative, hydrogen, partial positive. Nitrogen is going to pull some of that hydrogen towards us, so hydrogen is going to be partial positive, nitrogen is going to be partial negative. Now, because of that, we have that nice attraction. Thymine, this is one whole unit. This is called a compound. Adenine, another compound. And this bonding between two compounds, it's called an intermolecular bond. Inter meaning between. And so there's that hydrogen bonding. Cytosine and guanine, they have 3 different regions where they can hydrogen bond. And so thymine and adenine, you always see 2 hydrogen bonds. Guanine and cytosine, you'll see 3 hydrogen bonds. And that's why here, all these small dots, that's why you see these 3. So 1, 2, 3 for these 3 hydrogen bonds. 1, 2 for those 2 hydrogen bonds. Now, any protein or structure that has high amount of cytosine and guanine, you can imagine will be more difficult to break apart because lots of these hydrogen bonds together. Okay, other things about these nucleic acids is that they replicate before the cell divides. In what are called somatic cells, these are cells of every cell in your body, so these are non-sex cells. These are, for example, in your skin, intestines, hair, eyes. All of these are going to carry the exact same DNA. Okay, and that's why if you notice your skin, you can have itchy skin. Your skin's always going to dry off. You're going to have some flakes of your skin coming off, and the cells are constantly replenishing themselves. And so each of your cells from your skin they're going to have the exact same type of DNA on them. Now, your sex cells, these are not somatic cells, and these are going to be found within your sperm and then also within the eggs. And these are all going to be genetically unique with their DNA. Okay, so a couple of differences. And why are these so important? Well, these cells carrying all this DNA will provide the blueprint of how you're going to be creating yourself. So once your cell says, I'm going to be a hair cell, it's always going to produce all the proteins needed for it to become a hair cell. Okay, so this is why it's so important. Looking at ribonucleic acids, what these do is that they're actually kind of the slave for DNA, meaning that they carry out DNA's instructions for protein synthesis. Okay, and because of that, it's going to have a complementary strand to one of the alpha helices of DNA. It's created from a template of DNA. It's organized by complementary bases to form a single-stranded helix. So here we notice one strand. What are the sugars? Sugars, we have ribose. And the bases we have differ. We have adenine, uracil, guanine, and cytosine. Okay. And there's 3 different types. They're all going to coordinate their efforts in order for us to duplicate our DNA to form our protein. Alright, so how do these complement to our DNA? Okay, so in our double helix, cytosine and guanine, color guard, right, always going to go together. AT, always going to go together. Cytosine, if our DNA has a guanine, it's going to bind to, it's going to have a complementary cytosine. So here's DNA, here's RNA. If we have a cytosine, we're going to have a guanine. Now if we have a thymine, we're going to have an adenine. But if we have a adenine here on our DNA, then our complementary is going to be uracil. Okay, so this is our RNA strand and this is our DNA strand. So these are the ones that might get a little confusing, but these are the only changes that you need to know for that. Okay, so here's that larger look again. And so if you look at this illustration, be able to describe the differences. Okay, so what's the difference between DNA and RNA? Look at the helices. How are these bond? What forms the backbone? So we have sugar and phosphate here. We also have sugar and phosphate for the backbone in our RNA. What will bind? Cytosine will bind to what? Guanine to what? And then you look at thymine and uracil and adenine. What do they bind to? Okay, and so that's it for chapter 2. Here's a review one more time of all the different organic compounds that we just looked at. And so what I want you to do is be able to define, looking at nucleic acids, look at one nucleotide, break this down into the different components, which ones are specific to RNA, DNA. High energy compounds, if you take ATP, what can you break them down into? Polypeptides, what are they composed of and what are they broken down into? Triglycerides, the different types, what are they broken down into? And then polysaccharides, are they utilized as is or do they have to be broken down into monosaccharides for their use? Okay, so I will see you in chapter 3.